Hey guys, welcome to Dr. Priyanka's Biology, where we discuss everything from biology to biotechnology. So today I'm here with the part 2 of our Operon series. In the first part, we have studied about what is an operon, what is the structure and component of an operon, and how it works, the types of operons. And today I'm here with the part 2, that is the lactose operon, popularly known as the Lac Operon. From the name we know that is Lac Operon or the lactose operon. It is something to do with the metabolism of lactose. So rather breakdown of lactose and uptake of this lactose into the cell. So that is the function of this lactose operon. And this was in particularly studied in E. coli, and we have already studied about the blue-white screening and all those components. So these are the practical applications of studying lactose operon. So now let's get started with this lactose operon and let us try to understand what is lactose operon and its functioning. So let's get started with lactose operon. So coming on to the definition of lactose operon. So what is this lactose operon all about? All right. So the definition says that it's a cluster of three structural genes. So before we move on to the definition of lactose operon, let us quickly recall the what are the components and structures of this operon. So the structure of an operon. Now, operon basically comprises of major components, that is the structural gene. Okay, so we have the structural genes here marked as A, B, and C. These are the structural genes. Then we have the regulatory gene which codes for the repressor. Okay, and we have the promoter and operator. Okay, so these are the components of an operon. So together these genes are going to code for the proteins, which in turn is going to allow or the carry out the metabolism of the biomolecule that we are studying. So this is the structure of an operon. So this was for a quick review or quick recall. Yes. So now let's try to understand the definition of operon. So operon in this case is the lactose operon that we are studying. It is also comprised of three structural genes which encode for proteins which are involved in the metabolism of a lactose. And along with this, there is a site on the DNA which is involved in regulation of the operon. Now this concept was proposed by Jacob and Monod and it was studied in E. coli. Now it is an example of an inducible operon. So we have already seen what is an inducible operon, and here there are two independent genetic systems which control the entire operon. The first one is the promoter operator, that is the leg Z, leg Y and leg A system, and second one is the promoter repressor, that is the promoter and leg I system. So these are the two systems which independently control the lactose operon. Okay, with this background, now let's go and see what are the components of a lac operon. Let's see what are the components of this lac operon. Now, this operon system, if you understand, if you see here, there is a girl who has turned her face away from all these other girls, right? Then there is a gate which needs to be opened. Okay, this is a gate which needs to be opened. Then here there are some friendly girls around. The first one is the promoter, that is saying hi. Then operator, that is also with a gesture of saying hi. That means they are inviting. And here there is a group of friends which together they are planning something. So this group of friends is the structural gene, and this girl who has turned her face away from all these other people is the repressor. Okay, she cannot get along with these people. Okay. Now, if the repressor is present, okay, it is encoded, it will go and say hi to the operator, right? So that means this promoter is blocked from making friendship with these structural genes. So exactly this thing happens when it comes to your operon. So if the repressor is not there, okay, if this girl doesn't come to this picture, so inducer will bind to the operator, so promoter will be free. Okay, here DNA polymerase will come and say hello to the promoter, and the doors of this school or this gate will open, and automatically the DNA polymerase is free to move towards this structural genes, and as a result the required proteins will be formed. So this is in general a little bit about the operon system. So now let us move on and understand what are the components of the Lac Operon or the lactose operon. So as in case of this picture, Lac Operon is also comprised of similar components. Let us see what are the components of the Lac Operon. So if you see this picture now, let us compare both the components now. So we know from the operon concept. Now here also the Lac Operon comprises of the structural genes which are marked by this yellow components. Then we have the regulatory region which are marked in green, and next is the diffusible regulatory proteins which are marked in this purple color. So here we have the operator and promoter. Okay, now this lactose operon has independent promoters. So one promoter is separate for the repressor that is marked by leg I, and one promoter is for the structural genes. Okay, so if you see here, this is a bunch of girls, so that is the structural genes. So we have the promoter and the operator. Then we have this repressor and the promoter for the repressor. Okay, and this is the structure of DNA which needs to be worked upon. So now there are three different structural proteins or structural genes involved over here, that is the leg Z, leg Y and leg A. These are the structural genes. Now each of this structural gene is responsible for producing proteins which will enhance or which will help lactose operon to be worked upon. So now let us see what are they encoding independently. The first one, that is B leg Z or leg Z, it encodes for a protein called as beta galactosidase enzyme. Now, the goal of this enzyme is to convert lactose into its components, that is the glucose and the galactose. So it codes for beta galactosidase, which is going to break down or metabolize lactose into glucose and galactose. Now, yet another important function the beta galactosidase enzyme has to play is the conversion of this lactose into something called as the allolactose. Okay, and this allolactose is the actual inducer which induces the Lac Operon. So these are the two functions of LAC-Z gene. So this we have to remember. Then next one is the LAC-Y. So the role of this LAC-Y gene is the production of the permease enzyme. Okay, it codes for the enzyme permease. From the name permease, that means it is something to do with the permeability. That means it is the enzyme which is responsible for the uptake of lactose into the cell. Okay, It allows the lactose from the culture medium to enter into the cell. So it has function of permeability. So that is the permease. And the next one is the LAC-A gene. So it codes for thiogalactoside transacetylase. Okay. It codes for thiogalactoside transacetylase. So its function is to transfer the acetyl-CoA, the acetyl group from acetyl-CoA to beta-galactoside. Okay. Although it does not have this particular gene or this particular enzyme, it does not have a direct role in Lac Operon, but it plays a some helping or the supporting role. It is a supporting or the side actor of the lactose operon. What it does is, okay, it detoxifies the thiogalactosides which comes along with the permease. Okay, with the action of permease, there is excess of thiogalactoside which comes into the cell. So it detoxifies this. So these are the functions of the lactose, structural genes of the lactose operon. Okay, so these things we have already seen the components. So I do not wish to go again and repeat the same things. So we'll move on to the next slide. Then again this particular slide we have already discussed the functions of individual components of the structural genes. So only extra two things that I would like to mention here, that the beta-galactoside, beta-galactosidase enzyme, it's a tetramer, and second one, that is the galactoside permease. It's also known as M protein and it is a monomer. So these are the things you need to remember. So let's move on to the second part, that is the regulatory proteins. Okay. So LEC-I, it encodes a regulatory protein called as the repressor. So from the name repressor, we know it is something that is blocking. Okay, it is repressing the function of something else. Okay. So on this repressor, okay, it binds to the operator site. Okay. So example we had seen of those girls, if you can remember this. So repressor. So this repressor, what it does is it goes and binds to the operator. Okay. It creates the structural changes there. So now this promoter, and it hinders the binding of the promoter and the DNA polymerase. So as a result, the structural genes cannot be transcribed. So that means it is not going to produce any of those proteins which are required for the lactose operon. Alright. So this is how these enzymes work. So next component is the promoter. So promoter is required for the RNA polymerase to come and bind so that there is initiation of transcription. And the next is the operator. So this operator region, it is required for the repressor protein. So when the repressor protein comes and binds to the operator, it is going to, it binds and regulates the transcription of the structural genes. So another point that we need to remember is the Lac Operon has three operator sites. One is the primary or called as the major one. And the second one, the next two, that is the operator site 2 and 3, they are the secondary ones. So these are little bit things that you need to remember for your NEET exams. So now let's move on to the next part and try to understand the inducers and the process of induction of Lac Operon or the lactose operon. So the term inducer, so normally when we are using inducer, now you will understand the term influencers better than the inducers. So influencers, when we are talking about social media platforms, Instagram, so there are something called as the influencers which influence the people, okay, and then they are like kind of an example, right? or they influence a process to happen, right? So these inducers, they work in a similar way, but they allow a process to happen, okay, or they facilitate the formation of some products. So in E. coli, normally these enzymes, okay, or the structural genes. So in E. coli, normally there is very little amount of this proteins encoded by the structural genes. But when lactose is available, okay, this entire process gets, enhanced, and these genes are produced, or these proteins are produced at much higher pace and higher amounts, and which further facilitates the breakdown of the lactose into glucose and galactose, right? Now these enzymes, they are kind of inducers, or they are inducible enzymes which get induced, okay, and the process they facilitate is the induction. Fine. Now the here how it works, okay. So even when, okay, this entire process is not on, or even before your induction, the cell will have its own store of beta galactosidase enzyme. So it will have a little bit of beta galactosidase enzyme. So we have already seen that the beta galactosidase also converts lactose into allolactose, right? So this beta galactosidase, which is already present in the cell, it is going to produce, or it converts the lactose into allolactose, and these allolactose in turn acts as an inducer and turns on the transcription of those structural genes. So here your allolactose is the inducer. So there is yet another example of inducer is the IPTG, which is isopropyl thiogalactoside. Now this particular component or a molecule, if you remember, we have seen this in the blue-white screening. Okay. Now the point to be noted over here and to be remembered from your NEET exams point of view is that this IPTG cannot be metabolized. Okay, so it is not used up by the cell, whereas your lactose is used up by the cell. So this point you have to remember the non that it is not metabolized by the cell. Okay, so that makes it very suitable for the experimental studies wherein we are not interested in studying the metabolism process, but rather to see how it works, right? So now let us see what happens when the inducers are absent in the system. So this is a structural gene we have. So when inducers are absent, okay, your repressor will get activated, okay, it will form the active repressor, it will code for the active repressor because its promoter is independent, and this active repressor will go and bind onto the promoter, sorry, the operator. Because of this, the RNA polymerase enzyme, it cannot bind to the promoter, and as a result, the transcription of the structural genes is been blocked. So this is how, okay, the Lac Operon operates when there are no inducers. So this point we have to remember. So this what we have seen, the role of repressor, it is the negative control. It shows how the repressor, it is a regulatory protein, how it acts in a negative manner such that there is no transcription of the structural genes and Lac Operon is not being operated. Now this entire thing of the repressor, okay, how it works. It is not something permanent, okay. The binding of the operator and the repressor is not a permanent process. So it undergoes a cycle of binding and dissociation. It is like, on and off kind of relationship they have. So your RNA polymerase is a very smart guy. So it is on very, it's stringently it notices when the repressor is bound to the operon and when it is dissociated. The moment it comes to know that the a repressor is not bound to the operator, it quickly fits onto the promoter and it starts the process of transcription. So, in this cycle of on and off binding and dissociation of the repressor to the operator, RNA polymerase takes the advantage and it starts the process of transcription. So, in this way, there will be very small quantities of this regulatory proteins being formed. Okay, the products of regulatory protein, sorry, products of structural proteins are being formed. Okay, so this is one way it operates, all right. So this makes it a leaky expression. That means it is expression is for the short time. It will, the moment the repressor is bound, the process of transcription will stop. So that's why it is called as the leaky expression. This is yet another point which needs to be noted down for all the NEET aspirants and those people who are interested in the competitive exams. Alright. So here this leaky expression, it is going to produce a few molecules of these enzymes which are essential, okay, or which are required for the entry of the lactose into the cell. The moment lactose enters into the cell, okay, it will get converted to allolactose by the beta-galactosidase enzyme, which is the actual inducer of lactose operon. Okay, and now all is in the court of allolactose, which will go and perform its duty of inducer. So next part, now let us see what happens to the Lac Operon when the inducers are actually present into the cell. So first one, let us take the example of presence of lactose. Okay, now in presence of lactose, beta-galactosidase enzymes what we have, it is going to convert some amount of lactose into the allolactose, which is the actual inducer. Okay, now this allolactose, what it does is it goes and binds to the repressor. Okay, so now this is the perfect example of what we are talking about right now. So we have the repressor. Okay, repressor was encoded when the lactose was not there. Now lactose is there, right? Lactose is present. So what will happen? This lactose will go and bind to the active repressor. So because of this binding, there will be conformational change. Okay, there will be conformational change in the structure of the repressor. Now, because there is change in the structure, it is no longer active, right? So, as a result, the RNA polymerase can go and bind on to the promoter and confirm the transcription of the structural genes, which in turn will code for the beta-galactosidase, permease, and thiogalactoside transacetylase enzyme. So, this is how the role of inducer, that is the lactose, come into the picture. Alright. So now this RNA polymerase can go and bind to the promoter and transcribe the 3 genes. Now this slide onwards, it is going to be very important for the NEET aspirants and the competitive exam students. Now why I am saying this is very important, because this is going to be the favorite questions, and like somehow it is being asked every time in different formats. Okay, that is the relationship of glucose and the lactose operon. See, try to understand the concept of operon. This operon, it's like a energy saving mechanism which a cell has. So if your required molecule is present which can be taken up very easily, why it needs to spend its energy in converting something, right? So it is not needed. So rather it would look for some better options. So that is exactly what happens in an operon system. If a simple molecule is very easily available, it can be very well taken up. So that is what happens in case of your operon. So let us moving back to the slide. That is the regulation of Lac Operon in absence of glucose. Now this glucose, if it's in very less quantity. Now we have to understand one concept over here, that there is one enzyme called as adenylate cyclase. Okay, enzyme adenylate cyclase. Now this adenylate cyclase, what it does is, okay, it converts, okay, or it produces AMP from ATP. Okay, but when the glucose levels are low, okay, this adenylate cyclase is active and it produces this cyclic AMP from ATP. And this cyclic AMP, what it does is, it goes and binds to something called as CAP, that is the catabolite activator protein. So it forms a complex of cyclic AMP and CAP is formed, and it goes and binds to a specific site onto the DNA that is called as the CAP site. Okay. It goes and binds to the cap site, and this cap site, now further what happens is, it helps the RNA polymerase to come and bind onto the promoter and carry out the transcription. So when glucose is low and lactose is present, there is higher level of transcription that occurs, and there is more of the lactose operon in the functioning. Okay, so this slide is telling you the same story what I told you just now. Now, yes, another metabolite activator protein cap, also known as CRP, that is the cyclic AMP receptor protein. Okay, now this cap site is located upstream, 35 base pairs upstream of the promoter. And because of this, there is going to be almost 50-fold increase in the transcription of the structural genes. So this story we have already learned. Now this cyclic AMP, what we have, it is like a hunger signal. Okay, when an E. coli cell is present in the culture medium and it is hungry, so what it will do, because it is not getting enough of glucose, right? That's the food it is having, right? So when there is glucose, scarcity of glucose, it is going to signal that, okay, I'm hungry, by producing this cyclic AMP, and this cyclic AMP in turn, it is going to bind to the cap protein, okay, and this complex will go and sit onto the cap site, and it is going to allow the transcription. So this is how it works. So this catabolite activator protein, it need not be active all the time. It will be activated only if there is cyclic AMP being produced, and that is in turn cyclic AMP will be produced only when the cell is having less quota or less amount of glucose. So this relationship need to be fitted in your mind. Once it is fixed, it will be very easy for you to understand this concept. So the point to be remembered over here is that glucose and cyclic AMP, they are in inverse relationship. Higher the glucose, lower is going to be your cyclic AMP, and higher the cyclic AMP, lower will be the glucose. These are the things that you need to remember and be fitted in your mind. So now what happens to lac operon when there is high amount of glucose? So now that is just going to be opposite of what we have already studied. So that means if there is high amount of glucose, automatically your adenylate cyclase, okay. So now let's see what happens to lactose operon in presence of glucose. So we already know that when glucose is present, or when glucose is present in higher amount, it is going to block the enzyme adenylate cyclase, right? And as a result, okay, there will be reduction in the production of cyclic AMP from the ATP, right? So, in presence of glucose, the intercellular levels of cyclic AMP, they are going to fall, or it is going to reduce. Now, this catabolite receptor protein or the catabolite activator protein, it cannot bind to the lac promoter, and as a result, the Lac Operon, okay, is very weakly active. So even if lactose is present, it is not going to go and bind. Now you understand this. When you already have, so I'll give you one example. Okay, you had a very nice lunch full of whatever you like, okay, whatever you enjoy. Say there was heavy meal that you have taken, there is no space, okay, there is no space in your stomach, okay. So if somebody is coming and offering you, now let us try to correlate this concept with yet another thing. Suppose now you have come from somewhere outside and you are sitting in your relative's house, and if relative has given you, okay, given you a plate with a glass of nice chilled lemonade and one glass of warm water. So when you have lemonade, why would you go and drink that warm water, that too in summers? Exactly same thing is happening to E. coli. When it has easy work, that is glucose, glucose need not be broken down further. It can be utilized in simple way. So why it would go and take lactose when glucose is present? So exactly this is happening in your lactose operon in presence of glucose. So even when there is glucose present, lactose operon will be very weakly active even if the lactose is present. So next let us see what happens when the glucose is absent. So we have seen this case that glucose is absent. That means your adenylate cyclase will be very much active. It is going to produce, or it is going to convert ATP, okay, cyclic AMP. Let us see what happens to Lac Operon when there is no glucose. So we already know that when there is no glucose, adenylate cyclase will be at its best. It is going to convert the cyclic AMP from ATP, and now again it is available for the CRP to get bound to, and because of this, okay, though glucose is absent, but there is if suppose lactose is present, okay, this particular complex, that is cyclic AMP and CAP or the CRP complex, is going to form. It is going to go and bind to the promoter, and that is how your lactose operon will start functioning. Okay. Now here it is using lactose as an alternative source, carbon source, because if glucose is available, it is going to choose glucose rather than your lactose. So in the absence of lactose, here repressor will go and bind, and that's how your lactose operon will be inactive. Okay. So the entire play is about control and coordinated action of their structural genes, which will transcribe only if the glucose is absent and lactose is present. So this is the key, or this point we all need to remember. This last point. Now let us move on to the last part of the lactose operon. So this is a perfect example of negative and positive regulation. So negative control is when there is gene expression, when there is binding of the repressor, which is going to block the further transcription of structural genes, and positive control is the example when we are studying about the cyclic AMP. So thus this is a perfect example for both positive and negative control. Now this point also you need to remember for your NEET exam. Okay, that Lac Operon is the example of both positive as well as negative control. So I hope you have understood the entire concept of operon and Lac Operon. I will be back with the tryptophan operon in couple of days. So till then, keep studying, keep enjoying, and please like, share and subscribe our channel, that is Cosmic Edutech. Thank you.